That's really cool. Did you see the bathyform this morning where we chose the site, those kind of pockmark looking features?
Argus isn't going.
Yeah, I'm yanking on you.
Yeah, stop doing that.
Stop it.
Quit it.
So I'm going to just keep stepping. So if you guys see
Yeah,
anything at
go
all
for
that
it.
makes you interested,
Oh.
you give a shout, okay?
Oh, you weren't talking to me.
I'm not talking to you. You just go along your heading until told otherwise.
Aye, aye.
Those were great cores. Thank you, Bob.
Welcome.
Bridge nav. Another step, same bearing. Correct.
Yeah, I ended up saying silty, fluffy, light brown, maybe organic rich.
Okay.
For their
All right,
sediment
I'm going down.
descriptions.
You,
So I'll leave it alone.
Yeah, let's get to the rock. This stuff's yucky.
The sepias are cool, though. I mean, if I could see them through all the
Yeah.
dust that you're kicking around. Oh, look at that. Beautiful.
So an interesting thing about sea pens in Canada, they found out that some larval fish develop in between the tentacles, gaining protection because the tentacles sting.
Oh, wow.
I don't know that anybody's ever looked for larval fish in the sea pens here.
Must be so tiny.
They are.
I think there were redfish larvae.
In Canada?
In the the Gulf of Maine you're talking about. There was a paper a few years back in Peninsula.
But that's another type of supporting service, Devin.
Yep.
The system service. If you find larval fish hanging out in the corals or sea pens.
Oops.
What was it?
Eggs. What is that over on the
What is that
right?
critter?
Looks like
I
the
think
carcass
I think that's a fish that,
is being eaten.
yeah, well, he experienced the wonders of the thrusters.
Oh, poor
no.
thing.
Buddy.
Uh-oh.
You don't want to zoom on that, right?
No.
Bridge nav.
What else have we got?
How
Sea
about a
anemones?
40-meter step at 240?
Thorny heads?
Roger, thank you.
There's some small eel pout. So if in the kind of at the 4 o'clock position to the lasers, there's an eel pout, a long, skinny fish, just going out of the screen.
Yep. Need to. Are you recording all these, Devin?
Yeah.
Are we stopping to smell the roses, or are we just
that an eel
getting where
parrot?
we're going?
What?
We're getting until I
What
hear
was
a
that?
shout.
Eel parrot?
I'll type it in the chat.
You guys see the thing
Okay.
in Argus Cam? It looks like the character from Mr. Pac-Man. That, not him.
Eel pout.
They're like pink, reddish orange.
Okay,
Oh,
there's
like little
anemones.
jellyfish.
Are they jellies?
Yeah,
Jellies?
like there's one there. A little,
Yeah.
yeah, a little jellyfish.
Thorny head, Devin?
Got it.
Meredith says it's a snakehead eel pout.
Yep. What? Oh, boogers.
You want you to.
Can you hear me? I don't want to be heard.
That didn't work.
I know.
There's a umbalula right in front of us.
Yeah. Oh, we got the
Maybe I
porch.
should just
We got to
type
turn
them
the porch
into-
light off. That's what's going on.
Much better.
Oh, and we also need to do that. How about that?
No more junk.
Perfect. Perfect
Thank
in
you.
every way. Forest
Do you guys see anything rocky on the sonar up ahead, or?
Nope.
No, ma'am, not yet.
Which way are we?
How fast are we going? 0.3?
Let's see.
0.5?
I'm looking.
I can't zoom in on this.
Adjusting my eyeball.
I'm fine with going 0.5 over this
Okay.
if we're not going to stop and smell the roses.
Yeah, okay. Bridge nav. Can we bring the speed up to 0.5 knots? And you can add in another step as well, please. Same bearing.
Want to zoom in on that now?
Yep.
Quick zoom.
It's another sablefish?
Crab up there.
There's also some, it looks like Peninsula sea pens, very small ones here and there as well. So we have probably at least two types of sea pens in this little area.
Well, it's some type of spider crab. I don't know my crabs. Probably thodid.
Missing some legs.
Zoom in now.
Another sablefish.
You zoom out.
They're good size fish.
Some small gastropods as well, here and there.
I think we ended up cutting off poor Paul's description of the sediments.
Oh yeah,
Zoom in now.
we did. Did not get all the way to it.
Okay. Okay.
Peninsula.
Zoom out.
Yeah, so I think we've got at least 3 different types of sea pens here. We have Umbellula, Peninsula, and this Holypterus or Funiculi. We're not sure yet what it is.
There's a long whip one there. Or is that seagrass? No, it's kelp.
Yeah, seagrass and kelp,
Yeah.
or kelp.
It's a nice asteroid. Looks like it's having a meal.
I think that's
Is that the white star there? The white
The white starfish, all humped up over something.
That's cool.
not sure if that's Rathbanaster or something similar. Sun stars.
So this terrain tells us there's a lot of food and there's a pretty good current.
Bridge nav.
But not enough to
We
create
have another
sand.
step, same bearing, same speed. Correct, thank you.
Aaron, about how far have we traveled since we left our coring site?
About 90 meters.
Okay, thank you. Just getting a feel for the distance over time.
This feels like lightning speed to me right now.
One interesting note about the sea pens, the polyps when we first landed were all facing downslope, but a lot of these have the polyps facing upslope. Most of them have polyps on one side of the colony, which might suggest that currents are variable here, since they have different orientations. Right, yeah, light and variable. The witch eel.
There's a flatfish. Is that?
Yep, there's a long fish and then flatfish. There's a rock there with some
a rock
predation.
With asteroids and some other things on it. We should be working our way towards the base of the escarpment, and usually that means lots of food.
We want to do a round of introductions, or did we do that already?
We haven't.
Nope, we haven't done it yet. Lead the way, sir.
You want to start?
Sure. You can go ahead.
Okay.
It's your idea.
Yeah, sure. I'm not sitting a seat right now, but my name's Steve Oskovich. I'm the science data manager and lead data logger on this cruise, but I'm kind of sitting and helping the scientists in the back row right now. To my right, Kira, do you want to go next?
Sure.
Introduce yourself.
Yeah, this is Kira Mizell. I'm with the U.S. Geological Survey Marine Minerals Group, and I'm along on this cruise to help explain the phosphorites and ferromanganese crusts that we find. Excited to look at the mineralogy and geochemistry of those in relation to these cool biology findings that we're finding here.
Okay, I'm Lisa Levin, and I'm one of the lead scientists on this cruise. I'm at Scripps Institution of Oceanography at the University of California, San Diego. And I'm a deep sea biologist with a passion for all these creatures and communities and habitats and the services that they provide to people. So I'll be out here trying to make sure we characterize the communities, we're especially interested in the hard ground communities.
My name is Gabriel Castro. I'm also from the Scripps Institution of Oceanography, and I'm working with, I'm a postdoctoral researcher in the laboratory of Professor Paul Jensen, and I'm an organic chemist by training. I'm interested in the
snail or something?
compounds that
cool.
the different microbes we isolate from these environments might produce, because many of them could be used as medicines and other industrial applications.
Hi, this is Devin Vlock. I'm a graduate student at Scripps Institution of Oceanography at UC San Diego, and I'm working in Dr. Lisa Levin's lab, and this is my first foray into deep sea biology, so I'm super excited to be learning a lot here.
And you're a data logger.
And I'm data logging today.
Yep.
So we move into the front row. Erin Heffron, I'm navigator on this watch and also helping with mapping when we're not down at the seafloor.
Yeah, I'm Bob Waters. I'm the Herc pilot, ROV lead this leg. And then Kylie.
Hi, I'm Kylie. I'm the Argus pilot.
And she's wearing a beautiful mash of animal prints.
Some cheetah, some zebra.
If only the audience could see.
Animals, oh my.
And I'm Al Santos, lead video engineer on this leg and standing the watch on the 8 to 12.
Do you know what that red fish right there is, Lisa?
Thorny head?
Thorny head?
Short spine or long spine?
You've got to zoom in. I'm forgetting now. I think the short spine are the ones that are more likely to be in deep water, but both live together. One of them's more likely to be shallow. Forgotten which. I always do.
I think the short spines also are typically a
Blicky.
little bit larger.
Not a rock, but a blicky.
Oh, there's a anthoptylum, another sea pen. So that's at least 4 different species of sea pen.
Wow. Is that a high diversity for sea pens?
You know, I couldn't say one way or the other, but it's
Okay.
you know it's not insignificant to have multiple species of sea pens, but there's one dominant one, obviously. This long, thin, whip-like one.
Hi, Lisa. We have a question in the chat from Crispin Little. If we saw anything of particular interest in the seeps from the cruise up north last month.
I don't think I can answer that because I wasn't watching most of the time. But Andrew Thurber is in the chat. He maybe could answer that. There's definitely a lot of cool organisms when I did tune in, but I should let Andrew, who ran the cruises, chime in.
Bridge nav, another step, same bearing.
Okay, so thank you, Tom Ladig. The long spines are the ones that are characteristically deep most of their life, and the short spines can be there, but they also live in and migrate through shallower waters. So Steve, the new sea pen you just saw was anthoptylum?
Yeah, it looks like a big question mark. It has a big curl, high polyp density, and kind of it looks like a suction cup, but it actually goes into the sediment a bit. This genus also has members that are known as rock pens. We've been on the search for one for a couple of months now. We haven't found any conclusive evidence of a rock pen in the past few cruises, but we're keeping our eye out for Gary Williams at Cal Academy, who's describing possibly new species of rock pen in this area.
Does that mean you would want to collect one and see one?
If we see one on a rock, that would be a good indication that's a rock pen. But
okay. Beautiful spot. Lots and lots of life here.
Starting to get a little more slope here.
That was my next question.
hagfish.
Yep, contours definitely closer together.
Tree.
I wish we would just see something hard.
Hagfish. Definitely. Got it.
Oh, there's maybe one rock. Maybe. Oh, false alarm.
Yeah, there's a few rocks happening now.
Okay.
Good.
And it's getting steeper.
Do you want us to slow it down?
I'm not worried yet. No.
Yeah, back
quite
row, you
yet.
guys are okay? You want to slow down so that we can stop more easily, or are you happy to keep moving?
Well, I'm just thinking about officially starting a transect somewhere around here.
Okay.
And I guess, how do you do that? Do you just drop a tag as the start? And end?
Yeah, we drop a target to start, and you let us know how fast you want us to try to go and how far you want us to go.
Okay. I think this speed is fine.
Well, this is pretty fast.
Is it too
If
fast for
well,
transect?
I mean, if we get to the slope, it is, but I was just doing the 0.5 just because we were on flat bottom, but I don't know. How steep is it going to get?
I mean, the highest slopes are 30 degrees, so nothing crazy.
Okay.
So give it a go.
Yeah, we can just try it.
Okay. Is it a problem if we have to change speed during the transect? Is that one of the
No,
stipulations?
I don't think it's a problem.
All right.
We just need to know the start and the end. And I was kind of hoping
So
to wait to start till we got into some rocky terrain.
We can always
But,
do extra.
Yeah.
We don't mind.
We can. Okay, let's officially start one.
Okay. I'm going to drop a target.
The data logger
That's
can kind of note the name of this transect, however you want to call them.
transect 1.
Start
With
transect
the
1.
dive number, yeah. And we're starting at,
Bridge nav, another
we're
step,
still at
same bearing.
103.
No, that's the max. We're at
1,000.
942.
Oh, I've been writing the wrong one down then. So what was the depth at the push course? I've been reading the wrong
Those
one.
were 1003.
1003.
Okay. So it's definitely getting rockier,
Yeah. Nice.
little bits and pieces. And most of the rocks have little white dots on them. I can't actually tell what those are.
Yeah.
And were we doing 100-meter transects?
Yes.
Okay.
But the looks like there's a lot of life on each rock, doesn't it?
Yeah.
And a lot of sediment.
Might be some brachiopods I saw.
There's a couple of paired holothurians doing something. Okay, what do those rocks look like to you, Kara? Pieces of,
Yeah, they're covered in sediment quite a bit, so it's hard to see, but they look like they could be phosphorite, coated with some ferromanganese crust. It's hard to tell the exact color. We're a little far away, but it definitely looks like the biology does like those hard substrates sticking up out of the sediments.
yep, the Brazing, I think a Brazingid. It's hard to, I don't know. That asteroid is, maybe that's a Rathbun asteroid.
It's like a tennis ball.
Is that a sponge?
Yeah, there's a green sponge there. We've been calling those moon sponges. They kind of look cratery, and we collected a few up in Channel Islands and northwards.
Starting to get more rock.
Rocky
Definitely getting more
bits.
rocks.
Yay, rocky bits.
Yeah, definitely looks like pretty black. Probably ferromanganese coatings on these, if not nice thick crusts. Especially on these larger cobbles here. Don't really have that shiny texture that we're expecting from phosphorite deposits. But not quite as bumpy and botryoidal as crusts can be.
They were very smooth though.
Mm-hmm.
Sponge on the left. We were calling this sponge dirty barrel sponge for a while.
Bridge nav, another step, same
A
bearing.
glass sponge.
I mean, the rocks look very black.
Mm-hmm.
So that would be ferromanganese then, right?
Yeah, I think so. And in these little bit deeper depths, not super deep, but a little bit deeper and exposed chunks, definitely ferromanganese crust is a good candidate for mineral precipitation here.
Just curious, how many meters have we gone since we started?
We've gone about 48 meters.
Okay, so half the transect.
Halfway?
Yep.
So we're interested in living what's on these rocks, and I'm gonna guess that the rocks that are sitting in the middle of sediment like this might have a somewhat different fauna than the rocks that are just on rock.
Yeah.
Each rock looks like it's got a community that's developed on it. If we weren't doing the transect, I'd zoom in and take a look at what's living on these.
Yeah,
but,
definitely as soon as we finish. get a few
fish.
close zooms.
You know, when we finish that, it's just going to be sediment, right?
Yeah, exactly.
That's how it always pans out.
No.
We'll find more rocks.
Meredith wrote that the Laduncunia moon sponge is the group that the cancer drug was developed.
Yeah, there's another one on the 3 o'clock right there, that green blob sponge.
There we have sea urchins. This should be Strongylus centrotus fragilis. These pink urchins here on the left.
Yeah, it almost looks like they congregate around the side, the lower flanks of rocks, maybe catching a little extra current or shelter.
Or there something could have died there and they all went
Yeah.
over. They're good scavengers. They also aggregate on kelp sometimes.
Yeah, that's what we saw last cruise, was bunches of those
Bridge nav.
on kelp, drift kelp.
Can we do just a 10-meter step at 240, please? That's right.
There's a,
It's pretty.
those are a pair of sea cucumbers there, Solus.
Oh, wow.
I'll write that in there.
With all the tentacles sticking out?
Yeah, yeah. It's they're sessile sea cucumbers. They're quite abundant in this region at certain depths, and yeah, they cement themselves to the rock and put out those feeding tentacles.
So do they also have kind of a calcareous set of scales on the outside?
That's right. Yeah, they're
Yeah.
it's pretty rigid.
Or rocks. How are we doing on our transect? Are we?
Not too much more to go. 30 meters tops. Yeah, more like 20 meters.
Getting more excited about the rocks here.
Yeah.
They're all full of things. I want to zoom in on them and look at them.
Yeah, there's a lot of smaller things on there.
We like small things in
Yeah.
our lab. So for people just watching, it looks like we're going over a lot of sediment with a little rubble, but what people don't know is that there's thousands and thousands of animals down there. They're just small, living
right,
in the sediment and on the rocks.
I'm going to call that end transect. The ship just completed a move, so if you want to look around, we can, or we can continue on.
This the end of the transect?
Yep, end of transect.
Okay, 1728. Our depth is about 900 meters, right? Okay, so I think we'll keep going.
Okay.
We're moving towards our first waypoint, right?
Yep.
But if we see a pile of rocks with animals, I'd like to, up ahead, I'd like to slow down and zoom in on them.
Okay, no problem.
I'll,
So I'm going to keep the moves to 20 meters or less so that we can slow down more quickly. Bridge nav. Can we get a 20-meter step 240? Correct, thank you.
So in theory, we should have, we'll have to look at the backscatter, but
We're
this
on
should have
white
been
bits.
rocky.
Yeah.
Okay, maybe we can zoom in on these rocks, or are we too far over?
We got time.
Zoom in now. You looking for little things?
I'm just wanted to see what the white, all the white is. It almost looks like scars of something. Hard to tell what we're looking at. Like to see, what do you see, Kira, in terms of rock composition?
Yeah, it definitely looks like there's a ferromanganese patina on there. It's got that sheen, but I see some brown chunks, which could definitely be phosphorite as the underlying rock.
There's a lot of sediment cover on here, but there's some bryozoans on the exposed surface, and I can't tell if that's a tentacular crown of a worm right there. Should right here. Oops.
Oh, hello, friend.
Oh.
Well done.
There's foraminifera on there. What else do we see?
So this is
Not
about
too much, but that does look like it could be an amphoretid tentacles or something like that.
Yeah, I'm liking that brownish.
And then there's some hydroid thing there. I guess I need the drawing symbol.
Looks like a big chunk.
Are we getting, can we just get some pictures? Yeah. There's something.
Yeah, looks like
It's
an amphipod.
crowned down there.
There's an amphipod up in the center on a stalk of some type.
Oh yeah, that's the amphipod right there. And then some of this looks almost like encrusted sponge. Okay, I think we're good. We're not going to take this rock, and I'm not sure it would even fit in our inserts.
That's a big old slab.
Yeah,
It's a big rock.
That's a slab.
The slab, yeah.
Bridge nav, another step, same bearing.
Bob, you want a junk zoom, or you going to tilt up a bit?
I could tilt up.
Hey, Bob.
Mm.
When we go up slope, I pay in, like, you know, slowly the whole time. When we go down slope, we go down backwards? Is that right? Or am I am I confusing this with forklift training?
Yeah, I mean, the problem with it going down slope is you can land Argus on the hillside. So if I'm out in front, then yeah. It depends on how steep it is.
Gotcha.
I got a lot of people watching. Greg said that was a feather duster. That's the bellet under the rock. Crispin said we had a scale worm on top of the rock.
Did you change the gain on that at all?
Harry Wickstein pointed
Oh,
out
I
we had a
Did you
big
change the
shrimp
gain on the
to the right of the
sonar at all?
rock.
I didn't.
Mm-hmm.
I'm just asking, did you?
And then, but
Okay.
you see there's a
Because it just
question
got brighter.
for
I
you.
didn't know if you were messing
Yeah.
with the gain. Or if we were going to see some rocky bits at some point.
Yeah, we had a good question in the science chat about how to ID phosphorites instead of manganese oxides and ferromanganese crusts. And ferromanganese crusts tend to have a softer look, really bubbly textures that we call botryoidal. They tend to be more black. And those are coatings on other rocks, so they're incrustations. That's why they're called crusts. So phosphorites are a rock
Could we
type
zoom,
themselves.
please? Can we just zoom in a little bit as we go?
Oh, you want to just keep the zoom in a little bit?
Yeah, it
Yeah.
depends on if you need it out to
Nope,
see where you're
I
going.
can deal with it. Okay, zoom in a little, Alan. Just say when.
That's good.
Yeah, phosphorites tend to be more,
Bridge nav.
a little bit more brown, polished, rounded, kind of bulbous. There's so much iron and
Color
manganese in
page.
this water column that it's easy to always precipitate even a ferromanganese coating on a phosphorite rock. So it's not so easy to tell, but if you can get a nice polished looking surface or a more brownish colors.
Pilots, I just made a slight bearing adjustment to 230.
Okay, I will adjust. Thank you.
230?
Yes.
230.
Yeah, I would also say that if it's a really thick ferromanganese crust, it can be a little bit more uniform in texture because that crust has been growing slowly in a similar manner for a long time. And phosphorites, on the other hand, are replaced carbonate when phosphate goes in and replaces carbonate breccias and other kinds of sedimentary rocks, so they can be a lot more variable. So if you see lots of different colors, it's more likely to be
So
a phosphorite.
Long Holothurians are panicia. You can see in the chat spelled out there.
Yeah, I've got them.
Got 'em. I mean, I assume geologically you're not as interested in having these solo rocks picked up, or does it matter to you, Kara?
I mean, phosphorites the solo rocks are fine. They tend to be broken up over time in this region. For ferromanganese crusts, it's nice to get a nice thick one, and these look pretty small.
Bunch of these gastropods are Bathybembics.
Snails, yep. See all the snails here.
Bridge nav, another step, same bearing.
There's shrimp, pink things on that rock. Or Bathybembics.
Pretty cool. Little snail guy.
That polyp, that umbellula had really big yolky masses in the base of the polyp, indicative of
Its own?
yeah, they accumulate these opaque, sometimes pale or orange colored yolky egg masses.
Lots of brittle star and starfish activity on these rocks. And slabs. Look at them all the they're brittle stars. Over there, under there. Must be a good place to hide.
Yeah. So
See all the arms hanging out along the edges.
The contours are pretty steadily spaced from here to I, so I kind of suspect this is what it's going to look like through I. But then as you move up towards J, we're going to get tight again, so that might be more of a cliff a little bit.
Okay. Well, I'm wondering if we should start transect 2.
Whenever you're ready.
Now.
You want
Well,
another one?
Are we gonna do it low, or because I kind of need to
I
look,
see.
see
I see.
better.
We should probably zoom out just a tiny bit. We're not covering as much area here.
How much do you want to be zoomed out, Bob, for a transect?
I actually think this is fine.
Pilot,
I'll live with it.
you're okay?
Yeah.
All right. Data logger, we'll start transect there. Bridge nav, another step, same bearing.
That looks like plant debris on the ground there. Seagrass or something under there.
Yep. That forward lateral. Forward thrust, whatever it's called.
So when I drop targets, back row, do you need to see those target parameters for a while? Are you logging elsewhere?
You've been leaving them up long enough. Thank
Okay.
you. Yeah, a minute or so.
I'm okay. It's 2 megs. It's okay. I can live with it. Yeah. Just keep an eye on the humidity in that bottle. Yeah, it's really low.
You can see like every rock
Good.
has its own starfish. Or maybe I can't actually tell if those are brittle stars, big brittle stars. Look at them all on the rocks.
Yeah, I'm interested in some of the rocks really have kind of a yellowish looking, which could be phosphorite. There was a good question in the science chat about the age and origin of the phosphorite rocks in this area. So they're mostly Miocene deposits, which is about 23 million to 5 million years. And they were, the origin is buildup of a lot of phosphate in the deep ocean and low oxygen conditions during the Miocene that allowed phosphate to start replacing a lot of the carbonate in the sediments.
Ooh, that's a beautiful shot of lots of life. Sorry.
Nope.
Interrupt you.
That's all.
Just exclaiming it.
Got to do both.
And if you do want us to stop and zoom, just shout.
So
it's a transect, don't do that. You have to wait, sorry.
that's why I didn't say stop.
Happy somebody was paying attention. I forgot.
This type of terrain is interesting because it tells us which animals rely on hard substrates, which ones are only on the soft substrates, and which ones are on both.
Yeah, what are these 2B things? The kind of
Sea cucumbers.
Okay. Yeah, they seem
They're
to be on both.
Yep.
Yeah, when you get this really nice black color, that really looks like
So
ferromanganese.
what is this strange texture to the sediment there?
I don't know.
That
It almost
is, I
looks
mean,
like
Nice rock.
That looked like old seaweed to me.
seaweed?
Oh, I was looking at the rocks.
like
You were looking at
or like a skeleton, coral skeleton, sponge skeletons or something.
a nice block there. Bridge nav, another step, same bearing.
Yep, a Swiftia. Is that what that pink coral is?
I couldn't quite tell. It could have been a small bubblegum coral also.
Okay, and
So,
that type of crab is that a tanner crab that we just went over?
yeah, I think
Big
that was a
one?
tanner crab.
Just about halfway through transect.
We still have a ways to go to our first waypoint, right?
To our second one to
Or
I.
the second one.
Yeah, that is about 200 meters.
The currents can't be too strong here because there's such a layer of sediment over all the rocks.
A nice boulder.
Ferromanganese coated.
Yeah.
rocks.
They don't look like super thick.
Bridge nav, another step, same bearing.
So these areas are reflective on the backscatter maps, so that I'm guessing the sediment cover is not very thick. Oh, that's pretty.
Are the two laser dots 10 centimeters? Did I hear that?
Yep.
Okay. Thank you.
So all the animals we're seeing here are, kind of on the order of 10 centimeters or more. Lots of them. We would call those megafauna. But there's also loads of macrofauna and myofauna associated with the rocks and the sediments. Well, I'm less sure about the rocks because we haven't sampled them yet.
Look how rounded that one is in the upper right.
Very smooth. Oh, there's some more rocks off to the left. Each one seems to have one of those orange stars on them. Olivia. These rocks.
Yeah, you see that like golden layer at the bottom?
Mhm.
That's really
That could be
interesting.
phosphorates.
Yeah. It almost
Oh,
looks
look,
like
there's
iron.
another. The slabs show it clearly.
Yeah. That's very cool. Two-layered stratigraphy on that one.
Greg says there's a lot of orange featherworms on the rocks. I'm not sure I'm seeing them. Oh, some of those. Oh, there we go.
Bridge nav, another step, same bearing.
Is the little
Ooh,
triangle
that's a pretty rock.
on the map our waypoint, Erin?
The triangle that's selected right now is the end of our transect.
Okay.
The next waypoint's there. It's very
Ooh.
hard to see.
Yeah, that's far away.
It's
How
only
close are we
less than
to
200
the
meters now. It's not
end
terrible.
of the transect? Are we
Okay.
close?
Yeah, we are 30 meters off end of transect approximately. Maybe a little less. Maybe a lot less, 20 meters.
Looks like we could get a good rock collection here.
That one's a Bracingid right there.
Yep. So,
Yeah, absolutely.
what's that big white star, Steve?
Not sure about that one.
Steve, we need to get you a stool or something.
Oh, I got a chair over here.
Okay.
I'm fine. This one's I think this one's a goniasterid here.
And the coral? Swiftia?
That coral looks like Swiftia, yep.
That's the only coral whose name I know.
Get some bigger rocks.
Yeah, that's a nice rock. We're so close.
Oh,
to
so
transect.
all the orange are the feather duster worms, I guess, and they pull their tentacles in when we disturb them.
Oh, maybe zoom out a little bit.
Zoom
You're
out.
making me a little nervous.
Coming up, Bob.
There's a mushroom coral as well.
Okay, sorry.
Anthomastus.
That's a
That was-
Anthomastus.
I don't know if that was a real request. I was just a little scared.
Okay.
We're going
Are
to
we
little old lady in a walker speed.
This is a good.
I'll
This
try
would be a good
keep
spot
it
to
internalized
sample.
next time.
I'm wondering, are we near? Were we at the end of the transect yet?
We're really close. We are 9 meters, 10 meters off end to transect. You can call it. We could do another one if you want later.
I think
See what
there'll
happens.
still be
here.
rocks in 10 meters.
What?
There'll still be rocks in 10 meters.
Yeah, but as we climb, we're going into lower oxygen even. So there was a lot of good stuff on that last batch of rocks we just
We can also
passed.
go back. That's not a problem either. It's your
not-
dive. Go ahead, Rich.
There's a nice sponge over there.
Yep. See, and now it's more sediment cover, but the rocks will still be okay, I think.
You there yet?
All right, we are end of transect. The ship just finished a move, so it's a great time to look around or make decisions.
Yeah, let's slow down and look around a bit. Or even stop and look around.
Yep, ship
That's
stopped, so
possible.
plenty of time.
There actually is a bubblegum coral too at 3 o'clock. Small single stalk. Paragorgia possibly.
Zoom in now.
Image is really hot.
So you can see
You got more zoom?
we have. So which coral is this one, Steve?
This is a bubblegum coral.
This
So
is it?
yeah, it's probably Paragorgia or in that family of the Paragorgiidae. It's difficult, you know, when they're so small, but it's most likely Paragorgia
All right, good capture.
or a different, more rare genus, Cibogorgia, but I think it's probably Paragorgia.
So there's a sponge to the upper right that I think Paul is interested in. And I think what we maybe let's just zoom in on each of the main kinds of organisms that we see so we can get pictures of them before we do any collecting or anything.
So you want to zoom in on little stuff.
Yeah. Well, they're not little to us, but
Little to the ROV.
little to the
We need porch light. Can you turn on the porch light there?
And this looks like a very large slab, and I don't think we'll be able to sample
Yeah,
any rocks
definitely
here.
an outcrop. Anything below the outcrop is probably broken from that same outcrop. So
what about the solo rocks that we see here?
yeah, those are good too.
We zoom in now.
So that's an Anthomastus, that big pink thing.
Oh, that's very cool.
This
Wow.
this group Meredith reminded me that there has been recently revised and the new genus name is Heteropolypus.
Oh, hetero, what?
Yeah,
Polypus?
it's in the chat. Back at 1050 timestamp.
All right, good capture.
Okay.
Some bryozoans
It's a big one.
on the left-hand side.
Should I stay in tight?
Yeah, probably.
And actually there's a cup coral there too.
Yep.
Just in the upper right, around 2 o'clock.
So just below it would be the bryozoan.
That's right. Yep.
We know what that bryozoan is. I'm seeing really long tentacles, which suggests there's terabellid here somewhere. See all the little
Spaghetti worms.
transparent, yeah, spaghetti worm type tentacles. Oh, and there's maybe a big tube of one. These are busy rocks. There's definitely a lot of things on here.
Yeah, we're at about.
Is that a tunicate?
Is that the? Yeah, is that the guy that has the long tentacles? He looks really dirty. Sorry about the jerky zoom. When you get in really close, it's like
Everything hurts.
he doesn't look healthy.
Looks like a tunicate. Then there's all sorts of, all that little tube mesh is probably agglutinated protozoans, like phorium type things. See all the tubes there?
Mm-hmm.
You can use the telestrator if you want to circle
oh,
that.
yeah, good
and they
idea.
can see it up
Okay,
in the front
I
view.
think all this stuff is probably agglutinated protozoan.
You all the way in it now?
That's all my lens.
Yeah, get a photo.
Cool.
Okay,
Moving on?
let's back out and,
Okay, back out now?
and continue. I think what we'd like to do is find a place where there's a good pile of rocks that are loose enough to put into the bio box.
Did you see Greg did ask for a.
Oh, he wants a close-up of a feather worm in the bryozoan. I missed it.
Wait,
We'll have
like
to.
really in close?
Well, we're moving again.
Well,
We'll have to.
I can park.
Do you see where there is one?
Greg said
Zoom
it's
in?
okay. Never mind.
Oh, okay.
We'll just.
Okay.
You don't have to settle down. Can tell it's Mixicula.
Ready to carry on?
Yeah, but let's go very slowly. We're just looking for a place where we can actually collect the rocks.
Okay.
And this outcrop, I think.
All right, we'll keep the
Unless you
steps
think
really
they can be
small.
broken off. I don't think they can be.
Probably not. There might be a few.
Zoom in out. I mean, maybe something up there. I don't know how attached those are. But
Do you, I mean, is it worthwhile taking the manipulator and tapping them and seeing
Yep,
how
can do
attached
that.
they are?
you can do that.
Apparently there's some brachiopods here. Maybe the white shells we're seeing.
Hey, can you